Hello everyone. This is the third video in our tutorial series on using the query tool in the QSAR Toolbox, the human health example. If you haven't watched already, check out the tutorials Query Tool Ecotox example and Query Tool Environmental Fate example for an introduction to the query tool and its core features. In this video, we'll walk you through how to search for chemicals that are positive with respect to in vitro genetic toxicity, for example, have positive Ames data, and are in vivo negative carcinogens. Are there such cases? To start, we need to select the relevant databases. The easiest way to do this is to select the relevant databases corresponding to the endpoint tree positions carcinogenicity and genetic toxicity. To see the endpoint tree, we need at least one chemical to be added in the data matrix. So, go to the input, select the CAS number icon, enter any CAS number, for example 66251, or select from the list and load a substance on the data matrix. After that, go to the data module. First, unselect all here if there are any selections. Expand human health hazards level in the endpoint tree and select the cell corresponding to carcinogenicity level. This action will automatically highlight and reorganize the databases containing carcinogenicity data if the options here are grouped by data selected on the data matrix. Now, all relevant databases are at the top of the list, highlighted in green. Select the checkbox of data available, which automatically select all the green databases under it. We have completed the selection of the databases containing carcinogenicity data. Now select the cell corresponding to endpoint level genetic toxicity, and the list of databases is reordered. Click on the data available, and we are done. We need to return to the input module and click the query icon. Confirm by clicking yes on appeared message. Let's start with definition of positive Ames data. Go to the Data tab, expand Human Health Hazards, Genetic Toxicity, and select Gene Mutation. After that, go to Metadata, pick Type of Method from the drop-down menu, then click the Add button next to it. The type of method is in vitro. Select it and then click the Add button. Repeat the same steps for test type, which is bacterial reverse mutation assay, for example, Ames test. The test organism species in this case is Salmonella typhimurium. Do not forget to click the Add buttons in order to set the rule. The metadata are set. In the current example, we do not need to define numerical metadata, so we skip this section. Now we need to go to the data section. Because this is categorical type of data, the rows mean, minimal, and maximal data are irrelevant. To define positive result, open the drop-down menu of the units row and pick a scale gene mutation 1. After that, pick positive from the other drop-down menu. Click Add at the bottom to include this query in the query tree logic panel. Since a single chemical may have both positive and negative genotoxicity data, and our goal is to find chemicals with only positive results, we need to define this criterion explicitly. To do this, go to the unit section and from the drop-down list, select negative. Click the Add button to include this query in the query tree logic panel. Repeat the process for equivocal data. You will now have 3 separate queries. The first query is for the positive data. The second query is for the negative data, and third query is for the equivocal data. Combine the second and the third queries, negative and equivocal, using the OR logic operator. First, right-click on each of the queries and then select OR button. After that, select NOT button. Finally, combine NOT with the first query using logical operator and right-click on the first query to change its color to orange and then click End button. This configuration instructs the system to retrieve only the chemicals with positive Ames data, excluding any chemicals that have positive and negative or equivocal results. Now let's define the data related to carcinogenicity. Go back to Data tab and click Clear All button at the top. Expand Human Health Hazards, Carcinogenicity, and select Summary Carcinogenicity. Under Metadata, click the drop-down menu and select Test Organism Species. After that, click Add button next to it. Choose rat as a test organism from the corresponding menu. After that, click the Add button next to it. We do not need other metadata details here, so we are done with this section. Now go down to the data section, from where select unit carcinogenicity 1, and the first category that we select is negative. Click the Add at the bottom to include this query in the query tree logic panel. Since a single chemical may have both negative and positive carcinogenicity data, and our goal is to identify chemicals with only negative results, we need to define this condition explicitly. To do this, replace the negative result with positive. After that, click Add to include this query in the query tree logic panel. Then change the positive with equivocal and again add it to the panel. There are 3 more not connected queries. Right-click on each of the last 2 queries, positive and equivocal, and once the queries are orange, click the OR logic operator, then apply not. Finally, combine not with the first query. You need to right-click on the query to make it orange in order to combine with a not, and then to hit the and at the top. This setup instructs the system to retrieve only those chemicals with negative carcinogenicity data, excluding any that also have positive or equivocal results. To combine both in queries, right-click each inbox in the query tree to highlight them in red. Then, click the "and" button at the top to connect them. Now, click Execute at the bottom to run the search. The results will appear in the data matrix, showing all chemicals that match your criteria. The system automatically collects relevant experimental data for the identified chemicals. You can expand the human health hazards and the carcinogenicity and genetic toxicity levels and see more details for the collected data. Congratulations! You've now learned how to use the query tool to search for chemicals which have simultaneously positive genotoxicity data by Ames test and negative carcinogenicity data. More tutorials are on the way. Stay tuned. Contact QSAR Help Desk for technical questions. If your question concerns policy or regulatory acceptance of Toolbox results within EU regulations of ECHA's competence, please refer to the ECHA website directly.